our country is a tropical one, but due to the climate change, the rainfall pattern has been altered, and that causes stress such as drought to the crops, including the corn production. Hence, that became a problem in agriculture because corn, or also known as maize, is the second major cereal crop in the Philippines. To adapt to climate change, we need to improve the system development, survive the water stress it's facing. One of the ways to improve the corn yield is by improving the root system through interactions with soil microorganisms such as bacteria, but as well as through genetics and nutrition. In our study, we used rhizobacteria, which is isolated from Binangonan Rizal, and was tested in corn to determine its efficacy in promoting root and shoot growth. So why did we choose rhizobacteria? Rhizobacteria contains growth-promoting bacteria that help in root and shoot growth. These are the 1-aminocyclopropane, 1-carboxylate, or the AAC deaminase, the indole-triacetic acid, or the IAA, and phosphatase. It is also a good choice because the ACC deaminase it contains produces bacteria that promotes plant growth by decreasing the ethylene, which is a stress hormone, in various plant processes. The researchers also found that these bacteria can produce exopolysaccharide, or EPS, which protect microorganisms from water stresses.
Additionally, rhizobacteria have been tested in rice by Suranta et al. In 2018 and Olam Riwacho and Pampalona in 2019. It was also tested in corn by Beltran but only limited to one variety. So far, rhizobacteria have been proven effective. The question is, will the other varieties respond the same manner as the corn variety tested? This study generally determined how rhizobacterial inoculation affected the growth performance of different varieties of corn during the germination stage. Specifically, this study examined the effect of the inoculation of rhizobacteria in improving root morphology. Furthermore, the responses of different corn varieties to the rhizobacteria was compared in terms of number and length of nodal roots as well as the length and dry weight of the shoots. This study was conducted under laboratory conditions at the Philippine Rice Research Institute Central Experimentation in Maligaya, Science City of Muñoz, Nueva Ecija from October to November last year. The researchers mainly focused on determining the corn germination responses in terms of root and shoot growth to develop proof of concept for long-term greenhouse and field studies.
Our experiment was conducted in a petri dish lined with filter paper under laboratory conditions at the DA Crops Biotechnology Center of Philrice. We use an actinomycete with 98% probable identity of Streptomyces mutabilis and has been proven to produce PGBD compounds. As a recap, the growth-promoting compounds are ACC deaminase, indole-triacetic acid, and phosphatase. This study used 6 different varieties and were randomly chosen. The corn varieties are Lagkitan, DMI, CN1777, IPV-VAR5, CW1722, and NK306. Let's proceed to the experimental design and the layout of treatments. The legend contains the main plots and the subplots. The main plots are the inoculation of treatments. I sub 0 is without inoculant. I sub 1 is with inoculant during seed soaking, and I sub 2 is with inoculant during soaking and germination. V sub 1 to 6 are the varieties and the subplot. So here is the layout of treatments.
For our culture management and inoculation treatments, 100 grams of sterilized carrier were used to make Streptomyces mutabilis inoculant. Next, 1 milliliter of actively growing Streptomyces mutabilis was inoculated in 22 milliliter arginine glycerol broth and was cultured for 3 to 4 days. Then broth culture was done, which brought the soil moisture to approximately field capacity. Then 100 seeds of each corn variety were soaked in concentrated sulfuric acid because it increases germination. About 50 surface sterilized seeds from each corn variety were pre-soaked in a 7-day inoculant culture broth for 30 minutes prior to sowing in petri dish. In our first treatment, the tissue was wet with distilled water only. This served as the non-inoculated treatment. In our second inoculation treatment, the tissue was wet with distilled water only compared to the first treatment. This has an inoculant during soaking. In our third treatment, after sewing, inoculant was also used to wet the tissue paper such that the inoculation was done at soaking and during germination. In general, our data gathering, the measurement was done for 7 days after germination. We measured the nodal roots by counting all the nodal roots that emerged from the base of the stem. We then measured the root and shoot length by selecting 3 random seedlings from the base to the tip of the root and shoot by a metric ruler. Lastly, the shoot dry weight was measured after the measurements of each shoot length by cutting the shoots at the base, put in a coin envelope, and oven dry at 70 degrees Celsius for 48 hours prior to recording of the dry weight. Then the data analysis and interpretation. For our statistical analysis, we employed the statistical error or standard error.
This graph shows the effect of the inoculation treatments on the different varieties in terms of the number of nodal roots. Based from the result, the number of nodal roots were not affected by the different inoculation treatments. Now here's the graph for the mean root length. The mean root length is the ratio between the total root length and the number of nodal roots. On the mean root length, only CW1722, Lakitan, and NK306 significantly increased during the inoculation with only soaking. During the inoculation with both seed soaking and germination, the mean root length of the same varieties increased with the addition of the IPV bar. Next, here is the graph for the total root length. During the inoculation and soaking, only CN1777 and Lakitan had the total root length increase significantly, while inoculation and soaking and germination, all corn varieties except CW1722 increase in the total root length. According to T et al. 2016, the overall effect of the S. mutabilis and increasing root growth was caused by the increase in the mean root growth but not the number of roots, because S. mutabilis produces hormone that can stimulate root growth but may not promote the formation of new roots, which could explain why root number does not increase with inoculation.
For shoot growth responses, only the Lakitan and CN177 were the only ones who significantly increased during soaking. On the other hand, during both soaking and germination, all the corn varieties significantly increased. The Lakitan and NK306 had the highest increase, while CN177 had the lowest. The same trends in shoot dry weight can be also observed in shoot length responses. Overall, the inoculation with S. mutabilis promoted the shoot and root growth of different corn varieties used, especially during the inoculation was done during both soaking and inoculation. However, the responses differ. The reason for that is their genetic differences, as they have different genotypes, which influences on how they respond to corn. According to Bulgarelli and others, the others may have DNA that makes them more responsive to the inoculation of certain types of resin material, or that it enables them to produce more root growth promoting compounds in response to the inoculation.
Corn is the second largest grain crop here in the Philippines. Although climate change related factors like drought have an impact on how much it produces every year. So to improve corn adaptations to climate change is to enhance the root system using bacteria. That's why we studied the inoculation of S. mutabilis to different corn varieties in germination stage. So based on the results, we conclude that rhizobacteria have a positive impact on enhancing the root and shoot growth development. Second, it is important to identify the variety which responds the most. Lastly, Lakitan is the most responsive among all the different corn varieties, indicating that it is important to have a careful screening of different corn varieties to enhance the effect of the inoculation in promoting plant growth. For the recommendation, the rhizobacteria can be packed as a biofertilizer component, which when applied to corn, it may help to improve their growth. Second, it is important to carefully select a corn variety that is suitable and shows stronger effect on the inoculation. Third, there is an opportunity to enhance corn's response to inoculation through the process of breeding. Lastly, this study can be extended to other corn varieties to test their shoot and root growth responses, such as with the use of rhizobacteria